Here in chapter 11.7, we're going to be talking about gene regulation, and specifically gene regulation and expression in prokaryotes. So recall that in eukaryotes, we also have regulation of gene expression post-transcriptionally. So remember that after transcription, then we had that 5' cap that's added and that poly A tail, and then we had introns and exons. So we kind of cut out some of the pieces of that pre-mRNA, glued the pieces back together that we needed or wanted for that particular protein, and that's what left the nucleus and went out to the ribosome. We are going to start by talking about prokaryotes and their gene expression, how that's going to be regulated at transcription. So in prokaryotes, our gene expression is regulated at transcription. So we're going to take a look at how it's regulated at transcription. And so recall that when we spoke about mRNA in prokaryotes, we said that within an mRNA, we don't have this excision and gluing back together. What we have is a whole bunch of different genes, if we're looking at the DNA before we get to the RNA, but a whole bunch of different genes within that section are going to encode for a whole bunch of different proteins. So the mRNA then is going to encode for those different proteins. And we talked about how that's very efficient, how we can make one long mRNA, and within that one long mRNA, we're going to be translating that into multiple proteins that all work together for some particular function. And the DNA that's associated with this is called an operon. So an operon is the section of DNA that contains these genes for some related function. So not only do they contain all of the genes for a related function or for related functions, they're also transcribed using a single promoter. So remember we have that promoter region that is upstream from this section, from these structural genes. And then we are going to talk about how that promoter works much more specifically here. But an example is called the Lac Operon, and that's one that you should be very familiar with. We're going to go through it. The Lac Operon contains all genes that are needed for utilizing lactose as an energy source. And so we see this in E. coli, but this Lac Operon is a very widely and important operon. All right, so let's take a closer look at this Lac Operon. So this is an image from the text. So notice that this region up here is labeled as the operon here, and this is because we have these structural genes here, and so they have them labeled in the DNA as A, B, C, and D for simplification here. And then notice also that we have the promoter region that's upstream to these genes. So let's talk about this promoter and operator. So these are what are called the regulatory region. Region. So the regulatory region includes the promoter and the region around the promoter where transcription factors bind. And something that we find in this region is the operator, and you can see that up here. So I'm going to move this up a bit. There we go. So the operator. And so this region that's next to the promoter is the operator, and this is a sequence that's within the regulatory region. So we have our regulatory region here, we have the promoter, then here, separate from the operator. And the operator is important because it's going to interact with different molecules. So we actually have some regulatory information kind of going all the way up here. But what we are going to focus on here is the promoter, which we've already spoken about, which is where all of the enzymes are going to attach to then start going through transcription. But then now we're going to talk more specifically about the operator and some different terms or different molecules that interact with the operator. So one molecule that interacts with the operator is called a repressor. So the repressor is a molecule that suppresses transcription. So suppresses meaning decreases or slows down, right? So represses or suppresses, it's going to slow down transcription. Then we also have something called an activator. This is a molecule that enhances transcription. And then we have something called the inducer. And the inducer is a small molecule, so smaller than the repressor or the activator. So a small molecule that interacts with the repressor or activator. So if we go back up to taking a look at that image, we can move back up here. So we have our DNA up here. We have our regulatory region, which is kind of this whole section here. Some regulatory genes up here. We have the promoter, the operator. Then we have our structural genes here. This is the region, as you can see, that gets transcribed. So we have the promoter. We have our enzymes attached here. We have the repressor or the activator, and we have our inducer, which we'll talk about in more detail in just a moment. And then this whole thing is going to go through and it's going to do transcription. And so then when it transcribes, we have our mRNA. And then remember, within our mRNA, now we have the information for all 4 of these genes. And so then all 4 of these genes are going to be translated into all 4 of these proteins on that same mRNA. So what this allows for, again, this is really efficient, and it's called constitutively expressed. So it is constitutively expressed. And what that means is that it is transcribed and translated continuously. So some of these regions are constitutively expressed. So it's efficient in that it has all of these proteins being encoded for in one mRNA or one section of DNA that includes this regulatory region, and it regulates it for the entire region. And some of these things are constitutively expressed, meaning transcribed and translated continuously. And what this does is it provides the cell with constant levels of these protein products. So for example, with Lac Operon, if the E. coli is in a medium that contains lactose, then it's going to be expressing these all of the time because it wants to be metabolically active, utilizing the lactose so that it can make ATP and it can divide and live happily. We also see things that are constitutively expressed being proteins that are necessary for DNA replication, so that we can always be going through DNA replication and therefore division, things for repair and expression of DNA, and then also in enzymes that are involved in this kind of core metabolism, so metabolizing lactose or metabolizing glucose, for example. So then others are expressed only when needed based on particular circumstances. So if the cell then finds itself so for example, E. coli, if it's utilizing glucose and it's living happily in glucose, but then somehow it switches to be in an environment that has now lactose, then it might turn on that Lac Operon and then make the enzymes for lactose. And so these are regulated by these transcription factors. And the transcription factors are the repressors, the activators, and the inducers. These are our transcription factors. So let's take a look at different types of gene expression and different types of operons that are related to that. The first operon that we're going to look at is a repressible operon. All right, so a repressible operon is an operon that's controlled by the binding of repressors to those operator regions. So we just saw when we were taking a look at the RNA, the mRNA, we have that promoter region, we have the operator region, and then we have all of the information past that or more downstream that is going to eventually code for proteins. So when we are talking about a repressible operon, we're talking about a repressor, one of those inducible factors, is going to attach to that operator region. A good example of a repressible operon, and the example that you need to be very familiar with, is the Trp operon. So in E. coli, tryptophan, which is an amino acid, is encoded by a Trp operon. And so this contains the genes that encode the enzymes that are required for this biosynthetic pathway. So in order for this to happen, in order for tryptophan to be synthesized, we have to use those enzymes that are encoded in this Trp operon. So when the cell is in a situation where it has tryptophan in its environment, then it doesn't need to make tryptophan because it's getting it from its environment. It has plenty of tryptophan that's coming in. When this happens, then we can repress this operon because we don't need it anymore, because we don't need to be making tryptophan because we have plenty of it in the environment. So if the cell is going through and utilizing RNA polymerase and making these enzymes and all of this stuff to make tryptophan, this is a waste of the cell's energy. So instead, it's just going to utilize the tryptophan in its environment. So when tryptophan in the environment, however, then is low, then the operon is turned on. When tryptophan is high in the cell, 2 molecules of tryptophan are going to bind to the repressor. So if tryptophan is low, the operon is turned on. When tryptophan is high in the cell, so at this point what we're saying is the opposite now. We're looking at the opposite for a moment, and we're saying that in the environment we have lots and lots of tryptophan. Since we have lots and lots of tryptophan, we have lots of it inside the cell because it's coming into the cell and it doesn't need to utilize the tryptophan operon. So when it's high in the cell, 2 molecules of tryptophan are going to bind to the repressor. They're going to bind to the repressor. It's going to cause the repressor to change shape, and then that binds to the TRP operator. Then when that is bound to the TRP operator, it's going to block transcription from happening. So I'm going to write this out, and then we're going to take a look at the picture or the image from the text. So we have 2 molecules of tryptophan. They bind to the repressor. This is going to change the shape of the repressor, binds to the tryptophan operator, and then that's going to block transcription. All right, so let's take a look at that from the text. All right, so this is from the text. It says, in the absence of tryptophan, the TRP repressor dissociates from the operator and RNA synthesis proceeds at the top here. Now let's skip down to the bottom part where it says, when tryptophan is present, the TRP repressor binds to the operator and the RNA synthesis is blocked. This is what we had just spoken about. So when there's lots of tryptophan in the environment, that means the cell is getting plenty of tryptophan. It's bringing it into itself, and so it doesn't need to have this operon turned on. Because here we have lots of tryptophan. That tryptophan is going to be inside the cell. When there's high amounts, then we have 2 tryptophan here. It's 3-dimensional, so there's another one on the other side. And it's going to bind to the repressor. The repressor is going to change shape, and when it does this, then it's going to attach to the operator. Since this huge chunk of 2 tryptophan and a repressor are all attached to the operator, then our RNA polymerase is unable to move down this operon, and then we're not able to make RNA. So it's blocked. You can see that here. And so this is because we don't need to do this right now. This cell would be wasting energy if it was utilizing this operon, and so this is a repressed operon. It's turned off. Now, once the cell gets into a situation where, say, it's utilized all of the tryptophan in its environment, and now tryptophan is low, when tryptophan is low, then the cell is going to need these 2 tryptophan to build its proteins. So the 2 tryptophan are going to be removed from the repressor. The repressor is then going to switch its shape, change its shape. That makes it dissociate from the operator. The operator is now clear, so RNA polymerase can now move down this operon, and it's going to synthesize RNA from this particular operon, which is good because then it's going to be making all of those different pieces of this operon. So then it's turned on, but then as this operon is turned on and it's making tryptophan and tryptophan and tryptophan, if it starts going through and making lots and lots and lots and lots of tryptophan, then that can actually get so high in the cell that then it's going to attach to the repressor, change the shape. The repressor binds to the operator. It stops RNA polymerase from going down because there's plenty of tryptophan in the cell until the cell uses it all, and then it's going to release and then go back to turning it on. So this is a repressible operon. The binding of the end product binds to the repressor and stops or represses or turns off the operon. It blocks transcription from happening. The other type of operon that we are going to talk about is an inducible operon. In an inducible operon, this is only required when the substrate is available. So the example that we have for an inducible operon is also from E. coli, and this is called the Lac Operon, and you need to be familiar with this one as well. So the Lac Operon contains genes that encode enzymes that are required for the metabolism of lactose. So as we said, it's only required when the substrate is available. In this case, the substrate is lactose, so when lactose is available, this is when this operon will be turned on. So when lactose is available, it is converted into allolactose in the cell, and then that's going to bind to that repressor. So again, we're talking about a repressor. We're going to bind to that repressor, then that's going to change shape, and then it will fall off like we saw before. Change shape and then falls off. So in this case, when there are high amounts of the product or high amounts of the substrate, then it's going to bind to the repressor, and that binding is going to make it fall off of the operator. So let's take a look at that image. All right, so again, this is from the text. So at the top here it says, in the absence of lactose, the Lac repressor binds the operator and transcription is blocked. And then at the bottom, in the presence of lactose, the Lac repressor is released from the operator and transcription proceeds at a slow rate. So this is what we were saying before. When lactose is available, so we have lactose in this case, lactose is brought into the cell because lactose is part of its metabolism, right? It's going to be metabolizing lactose in order to make ATP. So it brings that lactose into the cell. That's going to be converted into allolactose in the cell. They just say lactose here, but allolactose in the cell, this is going to bind to the repressor, and then it changes shape and it falls off. So then this is going to allow RNA polymerase now to go down this operon and make the RNA go through transcription so that now it has these particular enzymes here that are going to be necessary in order to break down lactose and get the ATP from the lactose. So in this case, we want the operon turned on when that substrate's available. So when lactose is available, we want the Lac operon to be turned on. And so we can turn it on by binding that allolactose to the repressor. It changes shape, falls off, and that allows RNA polymerase to continue down the operon. Now up at the top then, it says in the absence of lactose. So if lactose is no longer there, say the cell has then utilized all of the lactose in the environment, and now it has to go for something else to metabolize for energy. Then when we don't have it, then we're not going to have this lactose here to bind to the repressor. If it's not bound to the repressor, then the repressor is going to attach to the operator again. So the repressor will attach to the operator, and since it's attached to the operator, we have this huge molecule attached here. Now RNA polymerase is not able to go down the Lac Operon. If it can't go down the Lac Operon, then it's not able to make these enzymes that are required for metabolism of lactose. And that's fine because we don't want them. We don't need them, and it would just be a waste of energy because there isn't any lactose in the environment. So at this point, we would say maybe E. coli is utilizing glucose. If it's utilizing glucose for its metabolism and not lactose because it's not present, then we don't want to make all of these enzymes that are going to allow E. coli to be utilizing lactose when there isn't lactose. It's just a waste of energy. So this is actually what happens with E. coli. So E. coli uses glucose as its primary energy source. And then when glucose is depleted, then it can switch into using lactose. And this is why we have the Lac Operon, and we see this as an inducible operon in E. coli because it's going to be utilizing glucose, so that repressor molecule is going to be there because it's not using lactose at that point. But then once glucose is depleted, lactose can be used. But it gets a slower, or it has a slower growth rate with lactose. So even if lactose is present in the environment, if glucose is present in the environment, it's going to be utilizing glucose for metabolism. So it's going to have glucose. Once that's gone, then it'll switch to lactose because at least it's still metabolizing and it's still growing even though the rate is lower. And it can do this utilizing an enzyme called enzyme 2A. So enzyme 2A or E2A. So enzyme 2A is going to switch from glucose metabolism to lactose metabolism. So this is what allows E. coli to switch. So it switches from glucose metabolism to lactose metabolism. So this is going to then allow us to turn on the Lac Operon. So when the cell is in a situation where it has a decrease in ATP, right? So a decrease in ATP. And the decrease in ATP is because glucose has been depleted, right? There's a decrease in glucose, so it's not making as much ATP. So the ATP levels are really low because glucose isn't coming into the cell. What's going to happen is this is going to phosphorylate the E2A. So that enzyme is going to become phosphorylated. So we're going to add a phosphate group to E2A. That phosphorylation of the E2A is going to activate Adenylyl Cyclase. So Adenylyl Cyclase. This is an enzyme. So when that phosphate group is attached to the E2A, then that's going to activate or turn on Adenylyl Cyclase. When Adenylyl Cyclase is turned on, what it does is it's going to convert the remaining ATP in the cell, and it's going to turn it into cAMP. So Adenylyl Cyclase is going to take ATP, the ATP that's left, right, because there's a decrease in ATP, but there's still ATP around. So it's going to utilize any ATP that's still around. The Adenylyl Cyclase is then going to convert that into cAMP, which stands for cyclic AMP. And remember, AMP is adenosine monophosphate versus adenosine triphosphate, ATP, which basically means that we're removing two phosphate groups, right? Because now it's mono versus tri. So then we're taking that ATP, the Adenylyl Cyclase is converting ATP into cAMP, which is removing those two phosphate groups, and then we're going to see an increase in cAMP in the cell. When we see an increase in cAMP in the cell, we're going to have high amounts of cAMP around. When we do, the cAMP is going to bind to our catabolite activator protein, also known as CAP or CAP. So we have high amounts of cAMP. That's going to, the cAMP is then going to bind to CAP. Then what we have is a cAMP plus CAP complex. So the cAMP plus CAP complex is then going to bind to the promoter region on the Lac Operon. So it binds to the promoter region of the Lac Operon. So this increases now the binding ability of RNA polymerase. So basically having this cAMP plus CAP complex on the end of the promoter is going to allow RNA polymerase to attach more easily to that RNA, or to that operon rather. And so it's going to allow it to attach more easily to that operon, and then that's going to initiate transcription. But we also have to have that lactose there to remove that repressor so that it can go. So if we are low in glucose, we can see this happen, right? So if we have a decrease in glucose, therefore a decrease in ATP, then we phosphorylate, add a phosphate group to E2A. Adding that phosphate group to E2A activates Adenylyl Cyclase. Adenylyl Cyclase is then going to take whatever ATP is left and convert it into CAMP. High levels of CAMP are now in the cell, which means that there's a greater chance that it will bind to something. It does. It binds to our catabolite activator protein, or CAP. When we have the CAMP plus CAP complex, this is going to bind to the promoter region of that Lac Operon. And then what this does is it increases the binding ability of RNA polymerase. So then this is going to initiate transcription. And remember that even if we can do this, so we can go through this whole process, if we have low amounts of ATP, we can create this CAMP plus CAP. It'll bind to the promoter region of the Lac Operon. But if lactose is not present, say we just had a dip in glucose, but lactose isn't even there, we are still going to have that repressor molecule on the operator. And we're going to take a look at all of this coming together in just a moment with an image from the text. So lactose must be present. And it has to be present so that it can remove that repressor. So it must be present for removal of repressor. All right, so let's take a look at the image from the text. All right, so in the upper left-hand corner over here, it says, in the absence of CAMP, CAP does not bind to the promoter. And so transcription occurs at a low rate, right? So at this point, what we're saying is we do not have CAMP. We have very low amounts of CAMP, which means we have high amounts of ATP. Because remember, if we have low amounts of ATP, then we have high amounts of CAMP because we have that conversion happening because we phosphorylate E2A. That phosphorylation activates adenylyl cyclase, and then adenylyl cyclase is going to take the ATP and convert it to CAMP, right? So if we have an absence of CAMP, that means our CAMP is low, which means our ATP is high. So likely we're utilizing glucose. This cell is utilizing glucose. There's a high amount of ATP. There's plenty of glucose around. And since there's a low amount of CAMP, we do not have the complex of CAMP plus CAP. So CAP doesn't bind to the promoter. It doesn't bind until it is combined with CAMP. So then we have RNA polymerase is able to go down the operator, in theory. But the reason I say in theory is because we can't forget about the repressor. So step by step here. In the next one down in here, it says in the presence of CAMP, right? So if we have low ATP, then we have high amount of CAMP. So if we have low ATP, likely we were running out of glucose. So then we have our phosphorylation of E2A. That phosphorylation is going to activate adenylyl cyclase. That activation of adenylyl cyclase is going to allow adenylyl cyclase to take ATP, convert it to CAMP, and now we have high amounts, or we have amounts of CAMP. So in the presence of CAMP, then CAP is going to bind to the promoter with that CAMP, and then it is going to increase the binding ability of that RNA polymerase. So you can see that here that we have our CAMP plus CAP complex that binds to the beginning here of the promoter and basically helps to grab on to the RNA polymerase to allow it to attach so that it can go down this operon and make these enzymes. So then if we look on the right-hand side here, we're putting this together with the repressor. So up in the top here, our CAMP plus CAP complex is going to stimulate RNA polymerase activity and increases RNA synthesis, right? So we already said that our RNA polymerase is now going to be able to go down this operon and make all of these enzymes, increases the RNA synthesis. So we do have to take into consideration the repressor, though. So it says, however, even in the presence of CAMP-CAP complex, RNA synthesis is blocked when the repressor is bound to the operator. So remember that this repressor, if it is bound to the operator, that means that there is no lactose. Because remember, when lactose is bound to the repressor, it changes shape and it falls off. And it falls off to allow RNA polymerase to go down this operon to make these enzymes that are going to help break down lactose for energy. So even if we have low amounts of ATP, which means we have high amounts of CAMP, then we have high amounts of this CAMP plus CAP complex, it will attach to the promoter. It will allow RNA polymerase to attach to this operon more easily or more tightly, but it can't go anywhere because we don't have lactose. So if we don't have lactose, this repressor is still present, which is blocking the path for RNA polymerase. So we have to have all of these things coming together perfectly in order for it to work, which of course it will if the situation is present. So if we have both glucose and lactose in the environment, E. coli is going to utilize the glucose until it's depleted. We have low ATP, then we increase the C-amp, which increases our C-amp plus cap. Our C-amp plus cap attaches to the promoter, which allows RNA polymerase to attach. At the same time, we already have lots of lactose in the environment, we're saying, and therefore lactose has bound to the repressor. The repressor falls off, and so RNA polymerase goes down the operon, and then it's going to transcribe these 3 enzymes, for example, that will then be translated into proteins or enzymes that are then going to be able to break down lactose for energy. So this is how E. coli is able to switch from utilizing glucose to utilizing lactose. And this is an important piece that you need to understand each of these steps because the Lac Operon is something that is utilized quite often in biotechnology. Also the Operon. So make sure that you are understanding both the Operon and the Lac Operon and understand the repressed operon and inducible operon. So now we're going to take a quick look at some higher levels of gene expression and regulation. So the first one is what's called a Regulan, and a Regulan is a group of operons that are all controlled simultaneously. So rather than looking specifically at the Lac Operon or specifically at the Operon, with a Regulan, this is an entire group of operons. And so they're all connected and they're all controlled simultaneously. A second type of higher level are called alarmones. And alarmones, they have the core root word here, alarm. And this is because these are things that are produced during stress. So we have small intracellular nucleotide derivatives. So they're small molecules, right? They're derivatives of nucleotides, right? So they're slightly different than regular nucleotides, and they're intracellular, meaning inside of the cell. So small intracellular nucleotide derivatives are produced during stress. So these small intracellular nucleotide derivatives are produced during stress. When these are produced, it changes which genes are expressed, and then it will stimulate specific stress response genes. So if the organism is trying to fight off a host defense system. So if we have a bacterium in our body or bacteria, lots of them in the body, and then our immune system is picked up on it and they start to attack that bacteria. The bacteria would be in a stressful situation, and then what it can do is it can create these small intracellular nucleotide derivatives, and then it will change what's expressed. So for example, it might upregulate its virulence in that case, so that because it's saying I'm not going down without a fight, basically. So it's in a stress situation, then it's going to increase its virulence to try to fight back the host immune system. And another way to regulate what genes are made or gene expression are through alternate sigma factors. Alternate sigma factors. So remember the sigma unit. Remember the sigma subunit when we talked about RNA polymerase and how there are the different subunits. And one of those subunits is the sigma subunit. And so we can change, or a cell can change the sigma subunit of RNA polymerase, and that's going to change which sigma factor it binds to. And a sigma factor is on a promoter region. So we have different promoter regions within different operons have different sigma factors. And so the sigma factor is within the actual operon itself. And so if the RNA polymerase has one particular sigma subunit, then it can only attach to the sigma factors that match with that subunit. So in this situation, it finds itself in, it's going to utilize different sigma factors or different sigma factors with the sigma subunit. So if the cell senses that there's some sort of environmental change, then it can degrade the current sigma factor that it's using, and it can express or make a new sigma factor. So then when that RNA polymerase complex comes together, enzyme comes together, it'll come together with a new sigma factor that is new based on the new environmental condition. And we see this when we see sporulation. So in our Bacillus and our Clostridium genus, then we see this switching. So again, remember, the cell senses an environmental change. This environmental change could say, like, this is really bad. It's getting, there's not enough moisture here. We've run out of glucose. We've run out of lactose. So what we need to do is we need to switch, and we need to start forming spores. So then a group of sigma factors are going to control the regulation of sporulation. And so this change happens, we have sporulation stimulating signals, and because of those sporulation stimulating signals, then this bacterium is going to start to make an entirely different group of sigma factors. And this group of sigma factors is going to then utilize their RNA polymerase enzyme with the different sigma factors to express a new operon. And those operons would be important for sporulation, for example. All right, so we are going to take a look at another method in a little bit more detail, and it's related to our tryptophan again. And that is called attenuation. So let's take a look at attenuation. So in order to really understand attenuation, I think it's important to remember that we have both transcription and translation occurring in prokaryotic organisms at the same time. So we're not going through transcription and then it's being modified and then it leaves the nucleus and goes out there and then is transcribed or is translated. In this case, we have transcription happening, and at the same time, remember, we have that mRNA kind of hanging out there, and translation is happening at the same time. So if we think about the tryptophan operon, so at the beginning, we have what's called a leader peptide, and this leader peptide is encoded by a TrpL. This over here, so TrpL. And then what happens is it transcribes this whole first section here, this TrpL section. It creates the mRNA for that, and then we have the ribosome attached to that mRNA. And that mRNA is going to start to go along and translate this leader peptide that has been encoded by this TrpL. Now, as it's moving along, we have what's called a Terminator Loop is created. And this is when we have a complementary binding within that RNA transcript. So remember, that's still attached. We have the DNA is still being transcribed by RNA polymerase, but we also have the ribosome going along that mRNA. As it's going along the mRNA, then it's going to start to go across this TrpL, so that's translated, but then there are 4 regions. There's, 1, 2, 3, and 4 as they're called. And so between regions 3 and 4, they actually are going to attach to each other. They're going to form hydrogen bonds because they have complementary base pairs. So between regions 3 and 4, we create this Terminator Loop. And then because this Terminator Loop is there, transcription is going to be terminated. On the other hand, when tryptophan is low, then our translation of TrpL is going to stall at region 1. And that's because TrpL needs 2 tryptophan to attach to it for it to continue. So if our tryptophan is low, our translation of TrpL stalls at region 1. And this is because it's waiting for Trp to bind. So waiting for Trp to bind. But since tryptophan is low, it's waiting a while. Since it's waiting a while in cell terms, then regions 2 and 3 are going to form an anti-terminator loop. So regions 2 and 3 form an anti-terminator loop. So obviously the opposite of what we just spoke about up here. So we have a terminator loop which terminates transcription. Down here we have an anti-terminator loop. And so when this anti-terminator loop is formed between regions 2 and 3, then RNA polymerase is able to continue on and transcribe that trip operon. So RNA polymerase transcribes the trip operon, which is exactly what we want, right? Because if our tryptophan is low, that means it's not getting it from its environment. So it's waiting for tryptophan to attach, and since it's just not there, then that stalling is going to allow, and the formation of this anti-terminator loop is going to allow RNA polymerase to continue transcribing, and therefore continue transcribing the trip operon, which is what we want, because then it's going to start making tryptophan. So I'm going to bring in the picture or the image from the text. All right, so this is the image from the text. I'm also going to have a video, but here on the left-hand side, we can see that we have our transcription occurring at the same time that we have translation occurring. So we have our DNA, we have our TRPL transcript here, or TRPL within our DNA that is then going to be in our mRNA. So we have our RNA polymerase here that's gone along the DNA here. It's encoded for our TRPL. It's made the mRNA. This is our region 1 and 2, and you can see over here on these other ones that there's region 3 and 4. So our ribosome is moving along that mRNA, and it's going to be translating these things. So remember, when we talk about a high level of tryptophan, then the translation of that leader peptide that's encoded by a TRPL is going to happen. So here's our TRPL here. And so our DNA is here, our RNA polymerase has gone along here, and then our ribosome is also here. And you can see that it has this polypeptide here, and it's going along, and it has gone through section 1 here and section 2. So it's gone through section 1 and section 2, so we have that translation of that leader peptide. But then we have this terminator stem loop that's going to form. The reason that this stem loop forms is because we have tryptophan in the environment, because we have a high level of tryptophan. It's going to bind to this leader peptide. It's required in here, right here. So we have our tryptophan codons, which code for tryptophan. Since they code for tryptophan and we have tryptophan, it's going to move, our ribosome proceeds, it's going to keep moving without a pause. It's not going to stall. And so it's going to bump into this terminator stem loop. When it bumps into this terminator stem loop, then our RNA polymerase is no longer going to go along here, or our ribosome is no longer going to go along, and it's not going to make the trip operon. It's not going to translate the trip operon because transcription has been terminated, because we don't need it, because we don't need these enzymes to make tryptophan, because we have it, because it's high levels. Down here, where we have our low levels of tryptophan, here's our TRPL, our RNA polymerase is over here. In this case, we have our trip codons here, and our ribosome is going to stall. And it's stalling because it's waiting for that tRNA to bring those tryptophan along so that it can put it into this growing polypeptide. Since it's not there, it's waiting, and since it's waiting, it's able to form this anti-terminator stem loop. So it's not going to stop RNA polymerase from transcribing the rest of this operon. Since it doesn't stop RNA polymerase from transcribing, then transcription is going to proceed, which means that this trip operon is going to be transcribed and then also translated. So that it's going to make those enzymes that are then going to make tryptophan, which is important because our tryptophan levels are low. And so then we need to make that tryptophan in order to make our polypeptides or make our enzymes. So this is, again, another way to regulate. So we already spoke about our Operon earlier when we talked about our Repressible Operon. So we have that, and we also have attenuation here. That's all going to make sure that everything is going to be very efficient, that the organism is not wasting any energy on making things that it doesn't need. So the last method of regulation that we are going to talk about for prokaryotes are called ribose switches. So ribose switches actually interact with attenuation in a way. So let's take a look at ribose switches. So in this case, what we're talking about is where we have these small molecules, small intracellular molecules. They're going to bind to what's called the ribose switch region on the mRNA. And these little molecules are going to bind to that region, and what it's going to do is it stabilizes specific RNA structures. So it's going to kind of shift the RNA, and it's going to switch whether or not translation or transcription is going to be turned on or turned off. So this is going to influence the completion of the mRNA molecule itself, so it's related to RNA polymerase, but then it's also going to determine whether or not the protein is made using that mRNA. So it's going to affect the ribosome as well. So let's take a look at the picture from the text. So here on the left, we are taking a look at how it affects RNA polymerase. So we have the DNA down here at the bottom. You can see that here. And our RNA polymerase has already gone along here and made this mRNA here. And so you can see the ribose switch region on the left. It makes this kind of lollipop structure, this stem loop structure. And then you can see the anti-terminator stem loop here. So we just spoke about the anti-terminator stem loop being formed. And then in this case, transcription is going to proceed because we have an anti-terminator, so it doesn't terminate transcription. So our RNA polymerase is going to continue on. So this is in the on position. Now, if we look below that, if we take this small intracellular molecule and it binds to the ribose switch region, note that up here the ribose switch region is this bluish color. Down here it attaches to this bluish colored region, and the way I kind of think of it is it takes all of this DNA and it's going to kind of scoop it over there because it pushes down on this region and it pulls the DNA to the left. When it pulls that DNA to the left, it forms this structure. And then instead of having this anti-terminator loop here with this purple region to the right of the anti-terminator loop, instead, this purple region here is going to become a terminator stem loop because everything is kind of shifted to the left and it's no longer the correct complementary base pairing here for the anti-terminator. Instead, we have this purple region that is base pairing, and so then we have a terminator stem loop. And so our RNA polymerase is no longer able to bind because it needs to bind to this purple region for it to continue on. So then in this case, it's turned off, and that's how a ribose switch works, with this small molecule that turns it on and turns it off. On the right-hand side, we can see something very similar, but it's related to the ribosome, so when we're actually doing translation in this case. So again, we see the ribose switch region here, we see this anti-terminator loop here, and then this kind of brownish region is the ribosome binding site. So the ribosome binding site is available in this case, and so our ribosome is able to bind, and then we're able to make our polypeptide. So this is turned on because translation is going to proceed because it can bind and it can move on. Now, if we take our small molecule again and we attach it to the ribose switch area, kind of pushes in again on this ribose switch area, pulling it along like a thread, the rest of the DNA with it. So then it forms, again, this type of structure here, and as it did this, it was pulling this brownish region toward the left as well, and then it makes this kind of terminator stem loop. Since it has turned this into that stem loop structure, now the ribosome can no longer bind to the ribosome binding site because the ribosome binding site is in the stem loop here. So it can't bind to the ribosome binding site and then continue on with translation. So in this case, it's turned off. So this is another method of regulation. So we have all of these methods of regulation within these prokaryotic organisms to turn things on and turn things off. So again, so that they're not wasting any energy because they're small and they need to make sure that they conserve their energy. So now we're going to talk a moment about some methods of regulation for eukaryotes very quickly. So when we talk about methods of regulation for eukaryotes, we're going to first talk about an enhancer. An enhancer is a DNA sequence that's going to promote transcription. So it enhances transcription. Within this enhancer region on the DNA, so it's a DNA sequence, within this enhancer region we have what are called distal control elements. And these are just little sections of the DNA that's within the enhancer. And what they do is that they have the activators are going to bind to these distal control elements. When these activators bind to the distal control elements, they're going to interact with different mediator proteins, they're going to interact with transcription factors, and then they're also going to interact with the promoter. So when it does this, it's basically kind of all coming together, pulling all of the correct things together in order to initiate or enhance transcription. All right, so then it's going to allow all of this to occur and enhance, and I'm going to bring in the image from the text so that we can take a look at this and see how it's kind of folding. All right, so you can see down here that we have our DNA bending protein that's going to allow this to happen on the left-hand side here. You can see the enhancer that we spoke about with our distal control elements, which are just these kind of chunks of DNA in here within the enhancer that allow the activators to bind to it, and then that's going to interact with these transcription factors and mediator proteins that are all going to then attach to the promoter. And when something's attached to the promoter, it's going to promote RNA polymerase binding. And so then when it does this, then we have all of these things coming together, and then it's going to be able to move down and it's going to be transcribing. All right, so I'm just going to very briefly mention some DNA level control of transcription. And this is for eukaryotes. So first we have epigenetic regulation, which is where the DNA or the histones are then going to be chemically modified to influence transcription. So this is given the certain circumstances that that organism finds itself in, then it's going to actually change chemically what DNA is available or change the histones to make certain DNA available, and it's going to be chemically modified to adjust transcription. And the next one is methylation of cytosine. So in response to environmental factors, the cytosine in the DNA, so the cytosine nucleotides, are going to be methylated. When they're methylated, then that's going to decrease gene expression. So we're not going to be able to go through transcription and then, of course, translation because of this methylation. So it's going to make it more difficult for us to express those genes. And lastly, the histones. So when we are talking about epigenetic regulation and modifying things, we can modify the histones by acetylating them. Acetylation and deacetylation is going to adjust how things are packaged. So remember, the histones are the proteins that DNA is wrapped around. So they can either be acetylated or deacetylated, and that's going to change how that DNA is wrapped up in there. So it's going to, as it says here, changes the availability of the loose DNA for transcription. So both of these, I think I might have said this in a little bit of a confusing way, are epigenetic regulation. So this chemically modifying DNA or chemically modifying histones are what we see here. So we can chemically modify DNA by methylating it, or we can chemically modify histones by acetylating it. And so this is going to be in response to environmental factors, and it's going to adjust what is available to go through transcription and then translation.